But it fit in really nicely with the assignment that I really wanted to bring to this class. It's one that I do in my coastal oceanography class that I really enjoyed, and I think the students really enjoyed. And I think because some of you have a lot more experience than my undergraduates do, I think that it'll work really well for this group. So the assignment, it's just a small assignment, and it's really an introduction to the phytoplankton in Harpswell Sound to get you thinking about who's out there, which organisms are there, when are they there, what are we going to see, and also for people who don't know much about phytoplankton, you'll start to learn a little bit about phytoplankton, and for people who have ever heard of an Imaging Flow CytoBot, or IFCB, how many people have heard of that? How many people have analyzed data from it? Any classifiers, phytoplankton classifiers? Okay, there we go. So there's a little something for everyone because we're all going to become classifiers. So what are phytoplankton? So this is from Iguana. This is a beautiful picture from the Plankton Journal, just showing you someone's artist's rendition of different groups. And so phytoplankton is literally from the Greek. It means drifting plants, and plankton means plants. They can't locomote. They can locomote. Some of them actually have flagella and they can swim, but they can't really swim against the currents, and that's what makes them plankton floaters. They come in all different shapes and sizes. You may or may not know the basic classification of them, but you can certainly appreciate how diverse they are in shape and size and color, which is a huge opportunity for us because if they are different in size and shape and color, that means they have different absorption and steric properties. And so that means we can detect those features from them optically and actually use that to classify them. Okay, so they come in all parts of the genetic diversity for all the organisms that are in the ocean. So if you're into that type of classification, there you go. That's as far as I'm going to go with that. So phytoplankton are obviously important because of photosynthesis. They draw down carbon dioxide. They create carbohydrate. That's not the only thing they create with their carbon. They make all the other body parts. And they also produce oxygen. They're the base of the food chain and are responsible for over 50% of the global primary production. So you breathe air. How many people breathe air? Yeah, thank your phytoplankton for the oxygen that you're breathing. They come in so many different shapes and colors and sizes. And that feature, in addition to impacting their optical properties, also helps define their role, their biogeochemical role in the ocean and their ecological role in their particular ecosystem. So that's really good for us because we would like to understand not only who's in the water, but once we know who's in the water, we can figure out what they're doing in the water. Are they responsible for which type of cycling, or who eats them, or who they actually, some of them actually eat other plankton. So they play a lot of roles. So the thing that I wanted to talk to kind of get a handle on today is size. And we're going to be talking a lot about size. Meg is going to be presenting size next week, this week? She's going to be talking a lot about size and size distributions. And so I want to start thinking about size. And we have some definitions. And for plankton, these are approximate definitions, but as long as we have some definitions, it will make it easier for us to have a conversation. So for some reason, everything is based on the number 2. So if something is less than 0.2, we tend to call it dissolved. Now that's really sloppy, particularly for the chemists that I see cringing, right? Why do we call it that? Because that's our filter size, right? It's just convention. If we had started off with 0.1 micron filter size, maybe it would be 0.1 would be dissolved. Or maybe we really wouldn't call it dissolved, right? Because we have colloids, we have complex molecules, we have lots of things that don't come into viruses, that don't come into chemically dissolved phase, but optically and operationally we treat them as dissolved. The plankton between 0.2 and 2 microns are the picoplankton, 2 to 20 is nanoplankton, 20 to 200 is microplankton, then meso, then macro. So phytoplankton tend to be in the sort of pico to micro size ranges, although you can always find exceptions. and find some like colonial, trichodesmium, or things like that, which will show up into these larger sizes. So Ivana said, well, okay, so if it was a little person, and that person was the equivalent of phytoplankton, the relationship between pico to nanoplankton is a little person to a whale, right? That's sort of the difference. Like when a phytoplankton is next to a nanoplankton, we're like, oh my God, it's a giant whale, right? So that's how the size differential feels. And then of course, if you're a microplankton, you're the Statue of Liberty. So when you stand next to the Statue of Liberty and look up, you're a picoplankton looking at a microplankton, right? So we tend to see them as like small spheres or something, but on their scale, they're not seeing that. They're seeing all the detail of the plankton around them. So just kind of put that in perspective when we're talking about 3 orders of magnitude. All right, so it turns out that if you look at the size, we can also begin to look at size as somewhat of an indicator of their classification in terms of how they make a living. Are they phototrophic? Are they heterotrophic? Are they bacteria, like, or decomposers? So we want to sort of think about, like, as our community structure changes size, it also is changing the biogeochemical role. If we have like really large phytoplankton, you tend to have a whole different ecosystem that results from starting with very large phytoplankton compared to communities that only have very small phytoplankton, which tends to be more recycling. So you can use size as sometimes a proxy for ecological structure as well. And so knowing who's there, knowing how big they are, and knowing about geochemical role, all those things are sort of interrelated. And so that's why we're really interested in classifying phytoplankton, because it tells us so much about how nutrients are recycled, how carbon is cycled, how carbon is transported, who's eating whom, and predator-prey relationships, et cetera. So this is why size becomes important and why classification becomes important. So I'm going to start with the flow cytobot. And Heidi Sosik, who is a dear friend of mine, and her former advisor, Rob Olson at WHOI, started with a benchtop flow cytometer, which is a instrument that starts off, they originally invented it to look at blood cells. And so they converted it to looking at phytoplankton. And so the basic premise of it, if you haven't seen it before, is that they use a process called hydrodynamic focusing, which basically means they're flowing a fluid, which they call a sheath fluid. They inject their sample into this laminar flow tube or fluid such that every cell gets aligned in single file, which is amazing because that means you can look at one cell at a time or one particle at a time. And so if you take this hydrodynamically focused parade of single cells, and so now we're going to look across this here. We're looking down on it. So here's the single cell. They're coming this way, flowing this way, surrounded by a sheath fluid. You can interrogate that with a laser beam, and then you can have detectors located all the way around that incident array. So you can measure the forward scatter, which we're going to find out forward scatter is dominated by diffraction and tells you a lot about size distribution of a particle. You can look at side scatter, which will tell you a lot about the composition and sometimes the shape of a particle. And you could also, if you were using a laser beam that was absorbed by phytoplankton, say in blue, you could measure the red fluorescence that results from that excitation. So for every cell, you're getting tremendous amounts of information. And you can always link it back to that original cell. So you start with a basic flow cytometer like this, and they optimized it for phytoplankton by selecting certain lasers and certain detectors. And then they made it, because who would want to take something like this and throw it in the water? They made it submersible, right? And it's big, but they got it in the water. So that then they could actually be interrogating cells basically continuously at the Martha's Vineyard Coastal Observatory and UCO. And they can look at the dynamics of single cell properties of side scatter, forward scatter, and fluorescence, which was great. They did some really great work doing that, but that wasn't enough. So then they were like, hey, why don't we actually use that signal, that fluorescence signal, which says, hey, that particle was a phytoplankton. Let's use that to trigger a CCD camera and take an image of it. So now in addition to also having forward scatter, side scatter fluorescence, sometimes two fluorescence signatures, we're also going to take an image of every single particle that's flowing through the system. So that's what they did, and that's the IFCB, the Imaging Flow CytoBot. So here's an example of some of the images that are captured. So it's imaging and flow, these particles are moving through. They're capturing these incredible images that you could take with a stationary microscope, right? If it's well focused. You can see the entirety of each cell. So that's cool enough in itself. And anyone who has seen the collection of images of phytoplankton from a sample, it's overwhelmingly beautiful to see the array of phytoplankton. And you guys are going to classify some, I can't, and that's great because you'll have one sample. Okay. But it collects a sample every 20 minutes because it takes 20 minutes to run a sample. So if you have a continuously running instrument, or say a dozen years of a continuously running instrument, do the math, 20 minutes sample, and maybe you're collecting, oh, I don't know, 10,000 images per sample, classifying by hand is tough, and that's if you know your phytoplankton. So what they did is they developed a classification scheme, an automated classification scheme. So you start off with the original images, then they do something called phase congruency, so they can begin to look at the continuity of interfaces. Edge detection, so you can see for each set of samples, now they're looking at just the edges of different phases within the cell, which would be the exterior walls, interior membranes. That leads them to what they call a blob processing. So, they have this, they can identify the whole exterior of the cell and then come up with a simplified exterior boundary where it gets rid of all the interior boundaries because really they just want to look at biovolume. Once you have this, then you can look at an array of different biometric characteristics such as major axis, minor axis. Does it have CT? Does it have, is it sharp edge? Is it round edge? So they originally came up with, I think, something like 270 different geometric features that they can measure based on this logometry. More recently, they realized that they don't need that. A lot of those are redundant and really don't provide them with any information about classification of phytoplankton. It's been really incredible work on this in the last couple of years. But essentially, they're using this, this analysis to classify them into different taxonomic groups. And so here's just an example from early work where they had an automated classifier and a manual classifier with a person looking at the images. And then here is the correlation between what the automated classifier thought it was versus what the person thought it was. And you can see that it's doing pretty well. There's a couple of cases where it might have identified something in the diddleum and it got identified as a pennate. They're both diatoms, I would be psyched about that. But you can see that they're able to do a really good job classifying compared to a person. And they also, when they're doing their work, they do, I don't know, maybe 10% manual classification just to check and make sure that the classifier is working. People are modifying this classifier for their specific needs, depending upon a region of interest or species of interest. So there's a ton of classifiers out there. They've now updated to a neural network approach, which is improving this even better than what they originally had, but it's actually quite amazing. So the data are shared on a dashboard. And so I have an IFCB, which normally is here, collecting samples in the Florida Seawater Lab in real time. A colleague of mine has his there right now because mine's stuck in England. Emmanuel, did you have one on Tara?
We have one on exports.
We had them on exports, but
Yeah, it's on Tara and the photo series.
Yep, so we had tar and coral storage. So there's all these different data sets, and Heidi Sosik is posting all of those data sets on her dashboard. So if you go to the dashboard, you can pick a data set right here. I picked Harpswell Sound. And so what you see here is wherever it's here, that's where we are. I don't know where it was. This was the 20th of January. I'm not sure why I picked that, but the 20th of January, here's the set of images from that sample. If you look on the time series, you can tell when I had it deployed. And so we had it a little bit in the beginning of the year, we had to go on a cruise, we had it, this part of the year, and then we had to go on a cruise. Then we had a nice long stretch. We had to switch instruments in here because it's on exports, but essentially the histogram here is of the number of regions of these images. Each one of these squares is called a ROE, region of interest. And so you can see approximately the number of ROEs will tell you something about whether you have a bloom or not. So you can look at that time series and say something about the biomass. Okay, so here's, so this particular sample has 3 pages or 3 mosaics of images, right? So, you can count them up and figure out how many. It tends to arrange them largest to smallest. So by the time you get to the last page, it's all the nanoplankton. It can detect down to about 5 microns, but it looks like a dot. So you can't really classify much below maybe 8 or 10 microns. But let's say we're really interested in this one. So we just click on that image and it pops it up for us. And it does 2 things. It gives us a scale bar, so we know how big it is. And it gives us the name of the file, which is the year, the month, the day, the time, hour, minute, second, which instrument measured it, and then of that sample, that unique sample, this is image number 2219. So every single image has its own URL, which is a way you can track things so that if someone's working on one of the same data set, you can actually say, oh, did you look at 2219? In this mosaic, everything is to scale. So if you see something that's small, it really is that size relative to that. So that's good. And you can figure out how big one is. So there's 10 microns. So you can really get a good sense of size. Okay, 10 microns. All right. So the other thing that you can look up, and you can just pull up a little plankton index. And this is just a set of images that they've collected from their IFCB. They have them by ciliates, coccolithophores, diatoms, and flagellates, etc. And it's really nice to be able to go in and look at images and be like, oh, I don't know, was it a Strombidium inclinatum or was it a Strombidium morphotype 1? Like, so you can look at this in real time and figure out who you're looking at. So for example, this is the one that I pulled off of that sample, and who do you think it is? I knew it was a diatom, so I helped you out there. And I went and I looked at this guide, and which one do you think it's most similar to? The what? Ladderia, right. And so why do you think it's Ladderia and not Bonardia? It's curved. So for example, the Bonardia, there's no sort of space. The cells are very square and connected all the way at their edges, whereas here you can see that they're curved. And so as you first look at these, and so here's another one, like what about this one and this one? What would be the difference between these here? Yeah.
They're internals.
Yeah, their chloroplasts are really differently arranged. And so when you first look at images, you're like, oh my God, they're all rectangular and stuck together. But as soon as you start looking at like, how would I distinguish this one from this one? Your eye will automatically collect on details that will allow you to separate them, right? So in fact, I would have guessed it was Ladderia also, but then I can check because if I go back to the dashboard, if you scroll down, there's this little thing called Autoclass, which means that they've run it through the classifier, which means if you click on it, you get a spreadsheet with all 3,000 images. And so we go there, it looks like this. So then you search for image 2219. Across the top are all of the potential groups that it could be classified into. And the numbers in the boxes tell you the probability that it matched that classification. So for example, there's a very, 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 very small chance that it could have been decalosome. But if you keep scrolling over, you see that there's a huge chance that it's Lauderia, right? A huge probability. So if there was something that you weren't certain of, you could go and look at the Autoclass and check that and look for the size, et cetera. So, yeah. All right, so these are two things that are very small, the 10 micron range, and they're blurry because I've blown them up. But one of them looks like a teardrop, and one of them looks sort of long, but you can see these shadows of something off to the side. They're going to be the tough ones, right? Because they're a bit blurry, they're small. So, I mean, you know, like you can go back to the thing and try to test yourself. I'm going to tell you these two because you will see a lot of them. This is a cryptomonad, it's a flagellate. And some of the samples that you'll be looking at are loaded with these, okay? They're small, but they've got that little teardrop shape. This one is actually a pennate diatom. I don't know what species, but I know it's a diatom because of its shape. And when you see some of the larger diatoms, you will immediately recognize it. It's important to separate diatoms from other small things. Some of the other sort of sub 10 micron cells are round and you're not going to be able to tell them apart. We call them unidentified or unclassified nanoplankton. They are important, but we have to put them into a size class that isn't classified by taxonomy. So, your challenge, you're going to work in 8 groups. You'll have either 2 or 3 people per group. Each group is going to receive the mosaic from a sample. Some of them have multiple pages, right? So this one happened to have 3. Some of them have more. That might be the group of 3. You're going to cut them out and sort them by hand into groups, okay? Then you're going to take each group and you're going to sort it by size, from smallest to largest. And then you're going to go around and talk to the other groups and say, did you get Lauderia? Did you get Lauderia? You didn't get Lauderia? Well, we have Lauderia, so you have to leave some space for Lauderia, even though you didn't have them. And you're going to make a histogram by pasting them onto a very giant sticky note, okay? So there's a whole lab assignment that is on the web page, I think, but I'm going to just put it up here so you can see it. The important thing is we have one a month. So it's close to December 1st at noon, January 1st at noon, February 1st at noon. I just pulled those images off. So some of you got a lot of pages, some of you didn't get very many. But the point is we want to compare to see what has happened over the last 8 months in Harpswell Sound in terms of species composition. So you're going to cut them out, and then, like, say you have a bunch of these. You will create a histogram. So you have number versus species, and species is sorted by size from small to large. You will take 2 squares, which is 2 inches, for each group, and you will just paste them. So if you have a lot of nanoplankton, they're going to be your smallest group. So you'll just, basically the nanoplankton, you're not going to cut out individually. You're just going to cut out 2-inch strips and paste them up. This one only has 1 eucampia, but it's big. It'll probably be somewhere over here. You'll just paste it down at the bottom. If you have a sample that doesn't have eucampia, you still have to have a spot for it with just nothing in it. The point is that then each of the 8 samples can be compared because they're going to have the exact same x-axis, which goes from small cells to large cells in a histogram. Okay? So you have your groups, but you're going to have to be talking to each other. And in fact, some groups have a lot more to cut out. So if you finished your 3 pages, please go and help them. And there will be things sorted out all over here. And at first it's frustrating. I am not going to pretend that it's not. But once you get it cut and sorted, when you start to lay it out and you have all of the 8 different months separated out, there's going to be a lot that we can do with this. And so I have on the assignment sheet like my goals. My goals are, number 1, think about phytoplankton. Think about who's out there and when you see them and when you don't. Think about their size. And if I said to you, convert this to biovolume, how would you go from a 2-dimensional image to a volume? And think about biovolume per mil, because this is a 5 mil sample that these come from. What if I said carbon? Could you give me a carbon distribution, right? You know something about the size. You could make an estimate of the 3rd dimension. You could come up with a conversion from volume to carbon, right? What if you think about who is eating them? Some of you are going to have samples that are mostly small cells compared to samples that are mostly large cells. What does that say about the grazers that are around? So this type of a size distribution by taxa can provide you with tremendous opportunity for interpreting ecology, interpreting cycles and flux and export. And starting tomorrow, we're going to talk about absorption and then scattering, all of which are really dependent upon size, color, cell wall, number of chloroplasts, etc. So then we'll be able to look at these size distributions and begin to think about how does that translate into absorption and scattering coefficients. So we're going to leave them up for the next couple of weeks. Once we have them, I'd like to see a version of them tomorrow, however far you get. But then we'll leave them up because we'll refer to them when Meg is talking about size. We'll refer to them when we're doing mean modeling. We'll refer to them when we're doing biogeographic cycling. And you'll have a sense of what you're going to see when we go out on the ship. And if you go to the real time, you'll see they only update it because it's not my site. They update it every week. Every week they have the new classification out there, but you'll be able to see who's out there now. And it's pretty interesting. Okay, you guys excited? I'm excited to see it. So there's 30 pages here. You should only need 8, but if you really screw up, just go ahead. We have 8 pairs of scissors. We've got 8 different groups. I'll let you guys self-organize. You can do that tonight. Everything's here that you need. Does everyone see what we're aiming for? Excellent. Cool. So this is already posted, I believe. Yes, it's already posted. So it has the links if you want to go and look at the dashboard and ensure yourself that I didn't pull off the 12 pages of mosaics for February, for example. But all right, that's your assignment for tonight. Have fun. Oh, and the other thing I think that's important is recognizing that an automated classifier is doing what you're doing. When you're trying to figure out who's who, it's also just doing a blob analysis. And it's like, if it's long and thin and has some things sticking out the side, it's probably a pennate diatom. But if it's blurry, it can make a mistake just as easily as you can. So it's also not being too enamored of a classifier and recognizing that manual annotation is really important. Right? Cool.
There's another tool that some of you may be aware of called Autoclass. It's a European tool that currently hosts a whole slew of images from different imaging systems, semi-ICD as well as scanners for zooplankton and a whole variety. And one of the advantages is you can have in one place both classification by many groups that you can look at, but you can also do biogeography. You can ask where in the globe has somebody seen this this one, because they all classify into the same one, and they're not classified really well, but they all do. And every classification has their name on it. So if somebody else sees that you classified, they might send you and say, it's this and not that. So there's some check within the community about classification, and people try to, there's some responsibility. But there's also the CTD, for many, the CTD data that was collected with maybe least data that was collected with that, there's for that as well in there. So you can start looking also at temperature range, So you need to name the death rate of that organism and where else in the world it's been seen. And one of the beauty of this is that these kind of data existed in the past with microscopy, but they were not easily comparable. Or getting the data is not easy. Nowadays, with the kind of computation power we have, you can see what the Japanese one are seeing in the spring, which is what you're seeing in some of the, in the opposite side of the world, around the Southern Ocean, and every nation goes and has periods there almost. You can compare it when you use the same method, you can compare it with what you see. And build a much more comprehensive picture than what you would get from what you do. That's key.
So one of the challenges you can imagine is NASA requires you to submit your data to Seabass, right? But NASA's used to radiance spectra, absorption spectra, maybe chlorophyll fluorescence, but they want these data because they pay for these data. So we've been working with them, Heidi has been working with a group of people to try to figure out how to submit this type of data to Seabass so that anyone could connect IFCB data to whatever optical data or remote sensing data that they have. And so it comes down to the fact that every single image has its own URL that has associated with it, not only where and when and what else was collected, but also what version of the classification was used, who, if anyone, did a manual classification on it. So the provenance of the data, or sort of the whole story of the data behind every single image. And so that has basically just been submitted. And so now anyone who has the type of data will now put that into Seabass and it will become part of the data that you can use. So you could just say, all I care about are diatoms. And you could pull off all the diatom data, or I just want this one species, or I only want things that are long and thin, or I only want round things. So you have a way of being able to sort through the Seabass data for that type of information as well, which is going to be really exciting. So I just want to remind you that you can go, if there's something that you're interested in that's not in Harpswell, you can go to the data sets, and those are the data sets. So you can see the exports data is there, the WHOI doc, names, the NDI Biological Lab, which is Mount Desert Island, they have one. There's salt pond. My husband is actually doing a project with these guys on the salt pond. So it's like, it's crazy. There's data sets. Almost everyone who has an IFCB puts their data sets here. It's all there for you. So,
and
yeah.
The other thing is, we have this long quest to try and get into this type of details, at least about functional type, functional group from space. If you look at the number of papers where they actually compare remote sensing with this type of data, they might be maybe 5.
Maybe.
Yet there's maybe over 100 papers about doing species or groups from space. So ask yourself always, what are they validating? What is used to validate? How did they decide it's that?
This fucosanthin as a pigment is not just diatoms. So people who've done it to HPLC have to be careful because pigments are not unique. Cells are unique. So hope you have fun with this, and I can't wait to see what your results are.
Sorry, so you said on the histogram the smallest would be nanoplankton?
Yeah.
So they don't have phytoplankton directly?
They don't have what, phytoplankton? No, because the resolution on the camera, 1 pixel is about 2 microns. So yeah, so they sort of set the limit. So reliably, you can see things. You can see maybe a cell that might be 5 microns, but it's just a sphere. Right? And so at 8 microns, you can start to see whether it's like got the tail on it, or if it's long and thin like a diatom, or if it's round. The only thing I would say with the nanoplankton is I think that you can sort out the pennate diatoms, and I think I would like you to do that. But if it's just, if you can't do anything else with it, that's fine, just put it in nanoplankton. Yeah. Which is interesting to even just compare the nanoplankton with all the microplankton. Yeah. So as long as you're kind of inconsistent, so just talk with each other, have fun, have some beers.
Yeah, there's beer.
So just like, I have 8, I think we bought out all the scissors from every grocery store. There's 8 sets of scissors, there's tape, just. Divide and conquer. Yeah. Fabulous. Yeah, last.
So 10 minutes, let's find our way to the lab. You should all be able to open it, but take your time, water, restroom, whatever you need.